Okay, good morning. Thank you all for joining us here today for the third session in our bibliometrics training series put on by the NIH Library. My name is Candace Norton. I'll be talking to you today about different tools and resources that are available to you to use when you're doing bibliometrics work. First reminder is for those who are in attendance live, please hold your questions until the end. Brief disclaimer on my part is to just let you know that this is intended to be a fairly comprehensive, high-level introduction to a variety of tools and resources. As the title of the course describes, it is a survey, which means that this is not an exhaustive list of all of the possible tools or resources that you could use to do this type of work. It is just to serve as an introduction to get you started. Also, please note that any specific record counts or links to access any of the tools and resources are current at the time of this recording. But if you are coming to us later, you should go directly to the source to confirm that the counts are accurate if you're citing them, or to confirm if the download links are still working. All right. So the objectives for this session are to identify some of the most common data sources for use in bibliometric analysis, in addition to learning about some of the tools that are available to you for data cleaning and processing, and also for data visualization. At the conclusion of this presentation, we'll also learn about some additional resources that are available to you for future independent investigation. So to start, we'll hear first about our data sources, because that's really where we begin with our process. We have to have some data to work with. So first up is Web of Science. This is a citation database available from Clarivate Analytics. This is a licensed resource, so if your institution does not license Web of Science, you will be unable to make use of this resource. But it is one of the primary data sources that are used in bibliometrics. Most frequently, we here at the NIH Library end up using the Web of Science core collection, which has over 73 million article records and over 1 billion cited references. The time scope for this resource goes as far back as 1900 up to the present. Web of Science includes over 20,000 scholarly journals in over 250 discipline areas, including the sciences, which includes biomedical resources and life sciences, as well as the social sciences and humanities. In addition to journals, the Web of Science core collection also includes data on books and conference proceedings. So when you're using Web of Science, one of the things that you can and should be aware of are the indexing that's available for each paper, because that will help determine what types of analyses you can do. So for Web of Science, you're going to be able to capture information on all of the authors of the papers, author affiliations, abstracts and keywords, if they're provided by the author, the funding information, and one of the most important pieces for us is that cited references option. That's what distinguishes a citation database from your standard bibliographic database, is that addition of the cited references being included. Additional metadata that Web of Science tries to capture whenever possible are ORCID identifiers, and then they have over 5,000 unified institution names. So if you're searching and you use the Organization Enhanced feature, which we'll be hearing about in later sessions, Web of Science has made a dedicated effort to go through and do some of the name cleaning on the institution names for you. But within Web of Science, you also have this Analyze Results feature, which can be used on any set of Web of Science results to help you gather some preliminary data about the set of records that you're working with. So some of the things you can look at are publication count by country or institution. You can also look at who's publishing in a particular topic area during a particular period of time. There's a lot of faceting and filtering that you can do with that Analyze Results feature right in the Web of Science interface. We'll explore that more in future sessions. So your results are also broken down by record count and percentage of total results, and give you the ability to have them sorted or ranked by field. Any analysis that you do can be exported or saved out of Web of Science. The things that you create and produce in Web of Science are not just limited to that interface. Anything that you do, you can export out to be used in other tools. Next up is InCites, which is another tool that's produced by Clarivate Analytics. It is based on Web of Science core collection data. It's actually comprised of 7 different editions of the Web of Science core collection. Their date range goes back to roughly 1980 to present. They're capturing, again, over 12,000 journals, 12,000 annual conferences, and over 50,000 books. InCites is not limited by the document types that they capture, so whatever is available to you through Web of Science is available to you in InCites as well. And again, the core discipline areas that are covered are the sciences, social sciences, and humanities. So some of the things that you would want to do with InCites is actually make use of the analytical features that are built in. So you can generate reports within InCites looking at different groupings of people or organizations. The one that we use most, or that I've used most in my work here so far, is the research areas. There's lots of different options available to you, so if InCites is a resource that your institution has, I encourage you to go in and click around in some of these different reporting options and see if they can be of use to you. Within InCites, you can also create visualizations and data tables. As with Web of Science, anything that you create in InCites can be exported into a format that can be used in another tool. One of the key things to point out with InCites as well is that you are able to set benchmarks for comparison. So if there's either another single comparable institution that you want to compare yourself with, or to generate benchmarks for your entire data set, you have that option. So listed on the screen are a couple of options for different types of reports that you can create right within InCites without having to do much extra customization work after. Another resource to point out is Essential Science Indicators, which is also produced by Clarivate. We're running through the Clarivate resources first. Again, this is another tool that requires a license to access. So the journals that are included in Essential Science Indicators are assigned to one of 22 research fields. So that's going to be a very high-level categorization. But if you need that smaller number of research fields for some reason, then this can be a really good system to work with. It does include multidisciplinary as an option. So if the documents that are included in that journal span across multiple category areas, it is then likely to be assigned to multidisciplinary. So keep that in mind if you're using this, that while that 22 can be of use to you in some contexts, it can also be fairly limiting in others. ESI reports are calculated on 10 years worth of data, so it's 10 from the present year back 10. So that is another thing to be aware of. It's designed to help you with more current work, less useful for more long-term or historical investigations. Within ESI, you can also view your results as a map view or the table. So once you're in there, again, look around, try running some different reports, see what's actually available to you, and what might be useful given your particular context. Okay, switching gears a little bit, we'll go back and talk about Scopus, which is another primary data source that is used commonly in doing bibliometrics. Scopus is produced by VOSviewer. It also requires a license to access. Most institutions that support this kind of work either use Web of Science or Scopus. Not everyone has both. Here at the NIH library, we do. So for us, it's a matter of choosing which data source has the information that we need, because Web of Science and Scopus each have things that they do better. And so with Scopus, again, over 71 million records, 64 million post-1970, including the references, and then over 6 million pre-1970 records as far back as 1788. In case you're trying to do any research that is very historical, this might be of use to you. Again, comparable focus to what we heard about in Web of Science in that there's a focus on the social sciences, arts, and humanities. Scopus does highlight science, technology, and medicine as some of their key subject areas. Within Scopus, you have access to site score metrics, SciMago journal rank, source normalized impact per paper, article level metrics via PubMed metrics. So when you're using Scopus, you get a lot of other metrics built in right when you're searching. Much like the other resources we've heard about, there are analytical tools built into the Scopus interface. A couple of ones to call out are that there's an author evaluator feature, which allows you to analyze an author's publishing output. So really getting down to that author level data that you might want. You're also able to compare journals. So putting in a range of journals that you're interested in, and then looking at a series of metrics as it applies to each of those journals. So gives you the option of doing some easy side-by-side comparisons. You're also able to search by document, author, the affiliation identifier, and then ORCID IDs. Scopus is updated daily and is also available via API. SciVal is another resource produced by Elsevier. Again, requires a license to access. Has over 38 million publication records and over 20,000 journals from 5,000 different publishers. SciVal is comparable to InCites in that it's providing that next level of analytical features on top of the core citations that you're able to get from Scopus. So within SciVal, you have the option of creating visualizations and these standardized reports. You're also able to set benchmarks within this tool as well. They like to highlight that you can identify and analyze a variety of collaboration opportunities using the features that they have built in. And then there's also a built-in topic prominence feature that allows you to evaluate topic momentum over time. Okay, so what do you do if you don't have access to Web of Science or Scopus? Not all institutions do. That was one of the limitations of me being able to start exploring bibliometrics prior to coming here. There is an additional resource on the market at this point that was released back in January of 2018, and that is Dimensions. It's produced by Digital Science. There is a basic version available at no cost for personal use, while also having an enhanced version, which is referred to as Dimensions Plus, that is available with a license. With the basic version, you can still do a fair amount of searching and get some, again, basic information, which will enable you to get a start in doing this. But the enhanced version is really where you get the most information. Yeah, so there was the update released in January 2018. Again, it's a fairly new player in the space. At this time, they have over 140 million publications, grants, policy data, and different metrics that are mentioned. So one of the things that's a little bit different about Dimensions versus some of the other resources that we've heard about so far is that their emphasis is really on the connections between your research objects. And so it's less about the single data source and more about how many different ways they can link you to different research objects that have that type of connection. So looking at ties between publications and grants, patents, clinical trials, and other alt metric mentions. Whether you're working with the basic or plus version of Dimensions, you do have full text searching available for over 50 million articles and books. This is made possible in part because they're able to access a variety of different open access collections and publicly available archives, including PubMed Central. So rather than what we typically expect when we're working with bibliographic databases and using a very refined controlled vocabulary or indexing system. Dimensions operates off of search filters and faceting, which is a different approach. They rely very heavily on AI and machine learning. So be aware of that when you're generating your searches if you use this tool. There's also an API available. And if you do have the benefit of using Dimensions Plus, much like with the other tools we talked about, there are visualization, benchmarking, and other analyses available in that platform. Unfortunately, those are not available in the basic option. You have to do all of that on your own. I have to talk about PubMed, which is produced by the National Library of Medicine. Over 28 million citations in PubMed from Medline, coming from primarily life science journals, but also books. Fields covered include biomedicine, health, life sciences, behavioral sciences, chemical sciences, bioengineering. This is provided at no cost to users. There's comprehensive coverage from 1966 to the present, with some select coverage of literature prior to 1966. Separate from the other resources that we've talked about to date, which are citation databases, PubMed is a bibliographic database, so it's not tracking and capturing the citation references, at least not in a way that the end users can make use of. All right, clinicaltrials.gov. So far we've talked about resources that rely predominantly on journal articles and information about articles. At this point, we're going to start looking at other data sources that might supplement the work that you're doing. So clinicaltrials.gov, another resource maintained by the National Library of Medicine, provides access to information on publicly and privately supported clinical trials on human subjects. Clinicaltrials.gov is a very rich resource because each of the trial records includes a variety of fields that you can search on, and it's still very reliant on text word searching within those fields, but there's a great amount of information available, and so it's worth your time if trials information is something that's of use to you. Particularly want to call out that the trial records include summary information, interventions, outcomes, typically include the protocol and adverse events. You can also download all of the content of clinicaltrials.gov for analysis. A few other sources of clinical trials data is the International Clinical Trials Registry platform, which is maintained by the World Health Organization. One of the great things about this is that this platform provides unambiguous trial identification. So if you have trials that are being conducted across multiple countries, you're able to have a consistent identifier regardless of where the trial's been registered. Additionally, the U.S. Department of Health and Human Services maintains a listing of other clinical trials registries. Could do an entire presentation on clinical trials sources. So if you're not able to find what you're looking for in clinicaltrials.gov or through the ICTRP, you may want to visit this listing of other clinical trials registries and see what you can find there. Okay, next up is the NIH Reporter, which is maintained by the NIH Office of Extramural Programs. Reporter stands for Research Portfolio Online Reporting Tools, and gives you access to different reports, data, and analyses of NIH research activities, both for the intramural research program and also extramural. This is a particularly useful tool for us here at the NIH, and perhaps for those of you who are working with grant-funded projects at your institutions. So in addition to capturing information about NIH-funded work, NIH Reporter also captures information from CDC, AHRQ, SAMHSA, as well as the U.S. Department of Veteran Affairs. The information that's available in Reporter is available to download for analysis with a feature called Exporter. So whatever work you do in the NIH Reporter interface, you can easily export and work with offline. In addition to the NIH Reporter, there's also Federal Reporter, which is maintained by Star Metrics on behalf of U.S. Department of Health and Human Services. This is another searchable database of scientific awards, but it's from additional government agencies beyond NIH and HHS. So when you're in this platform, you can actually search across different agencies by fiscal year, by the project leader and award recipient, also with some keyword searches across project titles, terms, or abstracts. And with Federal Exporter, you can download these data sets as well. So once you've become familiar with either NIH Reporter or Federal Reporter, it's fairly easy to go between the two systems. An additional resource to highlight is InCites, which is produced by the NIH Office of Portfolio Analysis. This is a resource that's freely available to users. It's a web application that provides bibliometric information for journal publications. So some of the types of information that you can use from InCites are the total number of articles within your analysis group, your mean number of articles published per year. It will give you your relative citation ratio, if that's something you're interested in. It will also give you the weighted RCR. Now, one of the main limitations with InCites is that you are limited to analyzing only the articles that appear in PubMed. So if you've generated your initial search in, say, Scopus or Web of Science, there are going to be some of your records that you can find within PubMed and some that aren't. So just be aware of that. InCites can be very useful, but that is a potential limitation. Patents. Again, this is an area that could have its own entire presentation, so this is very much just an introduction to possible sources for patent information. For those who are interested in U.S. patent information, you would want to go to the U.S. Patent and Trademark Office. So there's two different components to the searchable databases that they offer, and one is for patents that have been awarded and for patents that are still in the application phase. So be aware that if you were looking for patent information from the USPTO, you'll need to search both locations to be thorough. You can also go to lens.org, which is another resource that compiles patent information from multiple resources. The rest of the items on the list, starting with the European Patent Office all the way down through the United Kingdom Intellectual Property Office, are mostly broken out by specific countries. It is very challenging to find a centralized resource that adequately pulls in patent information from all of the different countries, because every country has their own patent office. Regardless of what they call it, it might be under a slightly different name, but each country has its own. And so to be thorough, you would want to be checking as many of these as possible as relevant to the request that you're working on. Okay, so that was our introduction to potential data sources. Next, we're going to start hearing about different tools that you can use for data cleaning and processing. Okay, the first step is back to basics with Excel. So most folks who work in any sort of office environment or library environment are familiar with Excel. So Excel is a program for creating, processing, analyzing, and visualizing tables of data. The perk at this point is that it's very familiar, it's readily accessible in most office environments, and people find it fairly easy to enter and view their data. If you're doing any calculations with your data, Excel is primed to do that for you. It's capable of doing basic arithmetic all the way up to more complicated functions and formulas. Depending on what data processing or data cleaning issue you're having, Excel may not be the easiest tool for resolving this, but there's usually a series of steps you can identify to work around that if Excel is what's available to you. Here are some of the examples of ways that you can use Excel to clean your messy data. Some of the more basic options include inserting and deleting additional rows and columns. which can be really helpful if you're merging and splitting your data into multiple columns. You want to have a place to put that data when you break it out. You can remove duplicate records. You are able to correct dates and times. And again, one of the more basic but really useful options is the find and replace feature. Again, all of this has to be done once you know that the issue exists. Excel may not be the most helpful in helping you identify problems that you don't already know about. But once you do know, it's fairly easy to make Excel work for you. OpenRefine is an additional tool for data cleaning, which is sort of a step up from Excel in that it can help you identify messy or incorrect data issues that you weren't aware of. So OpenRefine can clean your data, it can transform it from one format to another. And one of the great features is that it allows you to take your existing data set and actually go out to a variety of different web services and other external data sources and supplement your data set with any missing values. There's lots of resources available online that can help explain how to do that. Additionally, one of the more powerful features of OpenRefine is that it uses a selection of algorithms to cluster and merge values to facilitate name disambiguation, which we've heard about already, is one of the more time-consuming pieces of doing bibliometrics, is that the cleaner your data set, the more accurate your analysis. And so OpenRefine can really help cut down on the amount of time that you spend resolving some of the author names and affiliation names. Okay, so one feature that can be really helpful when working with OpenRefine is that it uses both rows and records. So this is most helpful if you have a multi-valued cell that you're splitting out, because then you can keep multiple rows assigned to the same record. OpenRefine is also very useful in resolving inconsistencies in the data set and can help you match your local data to other data sets. So one of the examples that's used fairly often for using OpenRefine with library data is if you're trying to capture your authority control headings for cataloging purposes. So the example I've seen most often is if you have your internal collection data and then link out to the Library of Congress subject heading and actually match up to make sure you have the appropriate authority control. Lots of other applications as well, that's just one example. Okay, so in addition to these two, we're starting fairly basic with Excel into something that's sort of a bridge between Excel with point and click and doing actual programming. We're moving now into talking about R and RStudio. So R is an open source programming language and an environment for conducting statistical computing and graphics work. So there is a steeper learning curve with R, but if you already have some familiarity with R, or if you have someone in your institution who does, there are many packages that are available to work with messy data. Since R is designed to do statistical computing, messy data is a reality of that work. So here on the screen are just a few of the different packages that are available. Okay, so Grid is another resource that's produced by Digital Science, the same producers of Dimensions, which we heard about a little bit earlier. This is another resource that's freely available to download. It provides an automatic disambiguation service to match author affiliation strings to institutions. So this can be really useful in that it has location-aware disambiguation. It recognizes multilingual names and different name variants. One of the things that has come up for me at least once or twice, and I'm sure for my colleagues many times, is that there are institutions with very similar names in countries whose primary language is not one that I'm familiar with. And despite excessive amounts of Googling to try to figure out if these places are the same, I just can't make that determination with confidence. Grid can be helpful with that. And because it's free, it can be a great resource for you. Okay, so at this point, we're talking a little bit more about data processing versus data cleaning. So at this point, we're going to start to hear about the Science of Science tool, otherwise referred to as Sci2. This is a tool that's designed by Indiana University and is also freely available to users. Sci2 is used for creating temporal, geospatial, topical, and network analyses and to prepare your data for visualization. So you're able to clean and analyze and visualize a variety of data formats because Sci2 has many built-in algorithms that you're able to access through a point-and-click interface. So it's a very powerful tool without requiring you to have extensive amounts of programming skills already. You do have to spend some time with the tool and make use of some of the training resources that are available in order to make sure that you're using the appropriate algorithms in the right way. But in terms of learning curve, it's fairly reasonable. Sci2 is able to support many different data formats when you import it. And one of the great things about it as well is that if you're doing the same general types of analyses month after month or quarter after quarter, Sci2 allows you to save your workflow, which can be great for your own reference, but also if you ever have to reproduce the work and show how you actually conducted your analysis to outside parties. Another tool in this space is VOSviewer. So VOSviewer is a software tool that was designed specifically for constructing and visualizing bibliometric networks produced by the Center for Technology and Science at Leiden University. Mostly hear about this tool in terms of its visualization capacities, and we'll talk about that in the next section. But right now, really want to focus on its potential uses for your data cleaning and preparation. One of the great things about VOSviewer is that in addition to readily accepting imports from Web of Science and Scopus and PubMed, which we talked about as some of our key data sources, you can also pull in records from Crossref and from EndNote libraries in your RIS format. Additionally, VOSviewer has the ability for you to create the source files, which can be constructed to facilitate your data cleaning using the two columns that are available. One is your label and the other is replace by. So depending on your context, this can either be a very powerful option because it gives you ultimate control over your name cleanup. It can also be somewhat time consuming depending on the volume of data you're working with because your thesaurus files must be created by you, the user. They're not automatically generated on your behalf. And this function allows you to either ignore or disregard relevant terms or to do sort of that find and replace feature, which will allow you to disambiguate any terms that are there. All right, next we'll start hearing about some of the resources that are available to us for data visualization. We're going to go back to Excel just for a moment because once you have your data cleaned up to a point where you're comfortable moving forward with your analysis, Excel can be very useful to you because there are a number of preset chart options available that allow you to easily preview what your data can look like in a variety of different chart formats. Now, of course, there's some guiding principles on which types of charts or graphs are most appropriate to represent which types of data. I will leave that up to you to make that determination. But just be aware that whatever Excel recommends as a default, you can customize, which is to your advantage. And the ways that you can do that are very quickly available through a few additional point-and-click menus. In addition to the base functionality that comes in Excel, There are a few additional add-ins that you can include with your Excel to give you some additional visualization options. A few of the add-ins are listed here. So Bubbles, GI Graph, Power Map, and People Graph. Depending on your institution, you may be able to add these yourself. You may have to get your IT administrator to assist you with that. Should you want to make use of any of these additional features, there's oodles of online resources available to you to explore those. Another tool that we use heavily here is Gephi, which is an open source software for network visualization and analysis. Again, this is another resource where no programming skills are required to use. Everything is done through point-and-click menus. There is still a bit of a learning curve. You have to know which menus to click, in what order, and with what judgments to make in order to properly generate your visualizations, but much less time-intensive than picking up a new programming language. Again, it's very useful for generating your collaboration networks and topic analysis graphs. There are many options available to you in terms of which layout algorithms you use, which will determine what your visualization looks like. There's also some built-in community detection algorithms and the ability to embed a timeline. So both of these features can be really valuable additions to your analysis depending on what your end goals are. From within Gephi, it's very easy to export a variety of file types. So you can export to an SVG file, which is a scalable vector graph. As we move on with our series, you'll learn about a couple of different tools that are available and how to use them to work with those file formats after exporting. Okay, we're back to VOSviewer again. So in addition to the data cleaning and processing features that we heard about just a few minutes ago, VOSviewer is primarily known for its ability to construct and visualize bibliometric networks specifically. So there's a very easy to use pop-up wizard that guides you through the steps to create your networks. Network options that are available within VOSviewer include those related to journal titles and other journal data, the individual researchers and their publications. You can do citation networks, bibliographic coupling, co-citation, or co-authorship relation type networks. There are some text mining functionalities built in, which will enable you to do word co-occurrence networks. Again, VOSviewer is really great in that it accepts immediate data imports from Web of Science exports, Scopus, PubMed, Crossref, and RIS format. Another tool, which is not one that we really use here, but is good to be aware of, is Cytoscape, which is another open source software platform for complex network analysis and visualization. Unlike VOSviewer, which was specifically designed to work with bibliometric data and visualization, Cytoscape was designed for use with molecular and genetic data, but over time has since expanded as people have recognized the value in creating these types of graphs for use. There are a variety of layout algorithms available within the tool, and their VisMapper feature is fairly easy to use and gives you ample opportunities to customize the details of your visualization. There are, again, ample training resources available online, and it's very easy to download and start using. And an additional tool, not to be confused with Cytoscape, which we just heard about, is CiteSpace, which is a Java application for detecting, visualizing, and analyzing trends in the scientific literature, primarily via network analysis. This resource was developed and is currently maintained by Chao-Mei Chen of Drexel University. It is being made available to users at no cost. One of the things that they like to highlight is that it's appropriate for use with very small data sets as well as large data sets. The largest that they have on record is over 80,000 records. The CiteSpace site has several training videos posted. And one of the features that I found particularly interesting about this tool compared to the others that we've heard about is that they actually have 5 built-in demo projects for you to work through in order to learn about all of the various configurations that are available to you. Okay, Tableau, which is an interactive data visualization and analytics tool, primarily known for its use in creating dashboards. Again, it's another resource that's very flexible in the types of data sources that it accepts. And they do have a tiered access model available, so regardless of what funding capacity you have at your institution, if you're not able to get access to Tableau desktop, if you're an academic institution, you might be able to reach out to Tableau and talk to them about their academic options. One thing to be aware of, if you are making use of their free option, which is Tableau Public, any visualizations that you create will be posted to their public archive of visualizations. So if there's something that should not be shared with the public, Tableau Public might not be for you. Again, due to its popularity, there are ample free training resources available online. And again, looping back to R and RStudio in terms of its visualization capacity. Again, there are several packages that are available to you. ggplot2 is one of the most popular packages for doing visualizations, but should also point out Plotly, Lattice, ggmap for doing any GIS type mapping, and also RColorBrewer. There are great features built in with the RColorBrewer package on applying colors to your visualizations. All right, and Inkscape. Inkscape is an open source vector graphics editor. So it's very similar to Adobe Illustrator, except it's open source, so it comes to users at no cost. It's most often used by us here to create graph legends and to tidy up any label placement on our network graphs. You can also use it for original design work, but for us in our workflow here, it is mostly to put the final polish on any visualizations that have been created before those get imported into our reporting template. Okay, so now we're moving on to the final section of our presentation where we hear about our additional resources. So beyond just the data sources that you need to be familiar with and knowing about what tools are available to you for your data cleaning, processing, and visualizing, there are some additional resources to be aware of for your own learning and understanding about bibliometrics and its potential and how to do it well. Again, this is not an exhaustive list. This is just an introductory list of resources that I personally found useful over the last year or so as I've been undertaking learning bibliometrics and have had some recommendations and input from my colleagues here as well. I'm not going to read all of these titles, but I would like to point out that the first two books on this list, Measuring Research, What Everyone Needs to Know, is a very small, lightweight book. It's very accessible and easy to read and gives you a lot of great foundational information. And then Meaningful Metrics, a 21st Century Librarian's Guide to Bibliometrics, Altmetrics, and Research Impact, is written specifically for librarians, by librarians doing this work. I have found that not everything in that title is directly applicable to the work that I do here, but again, it's a good introductory piece and is written in a very approachable style. Here's some additional books that have come out in the last few years. Again, because bibliometrics draws on other subject areas at times, so specifically pulling in network science in part. There are several resources here listed in that domain area. You don't need to read all of it necessarily, but they're good resources to have on hand if that's an area that you want to explore in more depth. And again, just a couple of slides of articles. For those of you who are following along with our training series, we have been providing recommended readings along with each of these training sessions. Many of these articles have been or will be included. These are just a sampling of what's available. So in addition to individual journal articles that cover topics relevant to the work that we're here learning about, there are also a handful of journals that you may want to monitor their table of contents. There's Science Metrics, Journal of Informametrics, Journal of Scientometric Research, the Journal of the American Society for Information Science and Technology, and the Journal of the Medical Library Association. The last two are more library focused, but they are quite common publication avenues for bibliometrics papers. For those who are interested in any blogs that may exist, blogging about bibliometrics is fairly sparse. So there is a great blog out of the UK, the Bibliomagician, and then CWTS from Leiden University. They're the creators of VOSviewer. They also maintain a blog that gets updated fairly regularly. And when we heard in our lesson last week about the Leiden Manifesto, that group also associated with Leiden University maintains a separate blog. For those who are Twitter users, that seems to be where most of the activity is in terms of social media interactions about bibliometrics. This is just a handful of the more active Twitter folks who are talking about bibliometrics. If you start following any of these people, you'll most likely see that they're in dialogue and conversation with some of these other folks, but also new names. You could create an entire Twitter feed of just bibliometrics. Outside of Twitter, the next most active space is still listservs and other online groups. Here is a list of some of the most active listservs. Not surprising, 3 of them are hosted by different library associations. And then there's also a Google group that's fairly active, as well as the LIS bibliometrics list out of the UK. Here are a few online resources. So in addition to the Leiden Manifesto and the DORA statement that have already been talked about in earlier sessions as part of this training series, there's also responsiblemetrics.org, which is a forum for discussion built on the UK's independent review of the role of metrics in research assessment and management. And then there's also the Metrics Toolkit, which is a fairly recent site that was launched in the last year or so, which provides you with evidence-based information about the various research metrics that are available across disciplines. So you can go to this site and type in the name of a metric or indicator that you've heard about, and it will give you a brief description about how it's used and how it's misused. If you're looking for additional conferences and trainings to attend, I have to highlight that the NIH Library, in conjunction with SLA Maryland, has produced the Bibliometrics and Research Assessment Symposium on a biannual basis here at the NIH main campus in Bethesda, Maryland. That event has been produced twice at this point in time, and I believe there are still conversations in the works about continuing to host it in the future. Stay tuned. In addition to that, there are week-long trainings available at the Center for Science and Technology Studies at Leiden University, which is located in the Netherlands. So many folks don't have international travel available to them for training purposes. So something to be aware of is that CWTS is partnering with Galter Health Sciences Library at Northwestern University to offer periodic trainings in the US. At this point, They are doing that twice annually, typically one session in the summer and one in the winter. And then you can also be on the lookout for continuing education sessions at major library conferences. In recent years, there have been sessions available from the American Library Association, the Special Libraries Association, and the Medical Libraries Association. In addition to that, there is a standing metrics special interest group meeting that is a pre-conference meeting that's available at ASIST every year. So thank you for following along with that very information-dense presentation. Again, just a reminder that this is intended to be a high-level introduction to a wide variety of tools and resources. It's really meant to be a starting point for you and not an exhaustive list. There's likely many other tools and resources that are out there that you could use. But for those who are just getting started, these are some of the things that I wish I'd had available in a nice, tidy package when I first started. All right.